It's now time to extend our thinking about linkage analysis a bit further. So to start this out, I want to just take a moment to think about, we're sort of now thinking about multiple genes simultaneously. So, genes A through G, for example. So just to reiterate, you can use the percentage of recombinants to tell how far apart two genes are on a chromosome, or if they're linked versus unlinked. Okay? So this might lead you to a question. If your maximum recombination rate is 50, what does that tell you about how long chromosomes are? So let's say we do a test, and we're going to test genes A and B, and we'll run a series of crosses and examine the offspring, and then try to figure out if these two genes are on the same chromosome or not. So let's say we set up a cross, and we get, I'm just going to shorthand through some of this. So then we're going to do the same type of cross where we're going to cross a heterozygote for A and B, so big A, big B, little b, with a homozygous recessive individual that is little a, little b, little b. So we expect to get a 1-to-1 to 1-to-1 ratio. And let's say we get something like this. Let's say we get 12 dominant, 268 dominant recessive, 197 recessive dominant, and 40 recessive. So in this case, for figuring out the recombination rate, what you want to do is take the small numbers and divide them by the total. Because in this case, these two bigger numbers, those are now the parentals. And in this case, the dominant, recessive are the recombinants. All right, so if we do this math, we have 517 individuals total. So 52 divided by 517 gives us a recombination rate of 0.1 or 10 centimorgans. So as with the previous example, A and B are 10 centimorgans apart. So let's say we now repeat this process with A and a new gene, C. And with a new gene, C, let's say we get a recombination rate of 15 in this case. So now what we can do is, if we think about picturing this actual chromosome, and here's gene A, and here is gene C. We know that these are 15 centimorgans apart. And we know that A and B are 10 centimorgans apart. And let's say that we also know that B versus C is 5 centimorgans apart. So what that tells us is if B is 5 away from C and B is 10 away from A, then B must be here in the middle between A and C. So if we do a whole series of these linkage analyses, we can move along the chromosome and start adding, mapping genes onto a chromosome. But you might have a question. Are chromosomes limited in how long they can get? So can you only have a chromosome that is 50 centimorgans long, right? Because the maximum recombination rate we could calculate would be 50. And the answer is no. Chromosomes can be longer than that. So you can have two genes. Let's say we have gene D down here, and let's say that gene D is 85 centimorgans away from gene A. So the way we'd have to figure that out is by linkage analysis between C and D and other genes that are in between here. And so what can happen is A and D can behave as though they are unlinked. So if we were to analyze this using a linkage analysis, we get a recombination rate of 50. So they're behaving as though they're unlinked because they're so far apart on a chromosome that there's almost always some kind of crossing over event that happens in between those two genes. So genes can be on the same chromosome but behave as though they are unlinked. And you can have, and in those cases, those genes would be more than 50 centimorgans apart from each other. And, why might this matter? Well, it's useful for getting an understanding of what traits have a chance of showing up together with each other. In the slide I'm showing you here, this is actually a picture of human chromosome number 11. And at the time this was made, we can't do test crosses or back crosses with humans. That's not ethical. So instead, the method we used to figure out which genes were located where on which chromosome involved really extensive pedigree analyses. Tracing different characteristics back through time to see where those characteristics tend to show up in tandem with each other. And so thanks to using that process and this type of thinking, you're able to pinpoint that all of these different conditions outlined on this slide come from genes that are all found together on human chromosome number 11. So this end result is then what's called a linkage map. And more recently, we've been able to combine this very old-fashioned technique of pedigree analysis and linkage analysis with genome sequencing methods. So we sequence a genome and then we can relate those sequences to what we know about genes from linkage maps to identify what sequences correspond with which specific genes. So we can then figure out the association between a genotype and a phenotype. And in spite of our best efforts doing this, so the linkage analysis has sort of fallen by the wayside right now, but there are still cases where there are disagreements between a genome sequence and outcomes from this type of linkage map. So we're still scratching our head over a number of these cases where we don't really know exactly what's going on. But clearly there's something going on that's shaping how traits get inherited. That means there's not always a one-to-one mapping between a genome and where we think those genes are on chromosomes, which is really kind of a fascinating idea. So I want to go back now to this fly linkage example where, so in a lecture quite a while ago, we talked about distinctions between, a genetic probability and, what the possibilities are, right? That was the bunny example. If you cross a, brown bunny with a white bunny, you have a certain expectation for what offspring would be produced. But in actuality, there's a lot of different possible combinations that you could wind up getting. So I want to ask the question here of if we take this fly cross, so here we're looking at, combinations of offspring. So either the dominant phenotype, dominant recessive phenotype, recessive dominant phenotype, and the recessive phenotype. And so here's what we got for offspring from that cross, right? 180, 22, 18, and 180. So 400 total. So I want to ask the question, could it be possible to get this type of outcome even if these genes are unlinked? So what I'm asking here is, how can we decide the chances of winding up with this type of outcome just by chance alone? And this is where statistics come into play. So I'm going to walk you through a statistic that we can use to answer this question of whether this outcome is likely just by chance alone. And I'll change possible here to likely. And the statistic that I'm going to introduce you to is called the chi-squared statistic. And this is a statistic that's a method for quantifying the difference between an observed outcome and an expected outcome. And so the basic rule of thumb for how this statistic is structured is that the larger the difference between an observed outcome and an expected outcome, the larger the chi-squared value that you'll wind up getting. And there's kind of two components to making this decision. The first step is to actually calculate a chi-squared statistic. And then the second step will be comparing that value to a critical chi-squared value. And the specific critical value will depend on the number of categories that are being examined, right? So if we had two categories, heads versus tails, and we flipped a coin 500 times and we got 498 heads and 1 tails, we'd probably start to have questions about that coin, right? And in this case, we have just 2 categories to consider. So for the question at hand, thinking about these different phenotypic outcomes, here we've got 4 different categories. And we'll come back to that point in a minute. Okay, so let's think about our expectation if these traits were unlinked. So if they're unlinked, then we would go back to expecting a 1-to-1-to-1-to-1 ratio, right? And so if we have 400 offspring total, then our expectation for each of these is going to be 1/4 times 400, or 100, right? Okay, and now the chi-squared statistic is calculated by taking, for each category, we'll use a fancy Greek letter for this, so we're going to add up across categories the number of observed individuals minus the number of expected individuals, quantity squared, divided by the number of expected individuals. Okay? And I like to kind of keep this organized in a table, just for simplicity's sake. So I'm going to calculate this chi-squared value for each of these 4 categories, and then I'm going to add up the total. Okay, and I have done the math, or have put some of that math onto the slide here for you. So for this uppermost category, that's going to be 180 minus 100, quantity squared, so 80 squared divided by 100. For the dominant recessive phenotype, that's 22 minus 100 squared divided by 100. For the recessive dominant phenotype, 18 minus 100 divided by squared divided by 100, and then recessive 180 minus 100 squared divided by 100. And to simplify the number crunching, what that will wind up giving us is a chi-squared number of 64, 60.84, 60.8, 67.2, and again 64. So if we add all of these together, we're going to wind up with a total chi-squared of 256.08. And let's just do the thought experiment now of if our observed perfectly matched our expected. So if that were the case, for the top category, that would be 100 minus 100, quantity squared over 100, so that's 0, right? So all of these would be 0. So if your expectation exactly matches what you get, then your chi-squared is going to be 0. And if your observed numbers are really different, then your chi-squared value is going to be huge. So this is starting to look huge, but how big does huge have to actually be? So that's where we come back to looking up a critical number. So in this case, we've got 4 different categories. And so for looking up our critical number, we take our number of categories minus 1. And that's referred to as our degrees of freedom. So in this case, our degrees of freedom is 3. And so now here is a table for looking up our chi-square value. So we've got 3 degrees of freedom, and if what we want to look at in this table is the value that is at this 0.05 column. So we're going to look at the 0.05 column, and so this is for a p-value of 0.05. So in the 0.05 column, the number that's listed in that part of the table is called our critical chi-squared value. And if our calculated chi-squared, let's just write this out. So if our calculated chi-squared value is bigger than the chi-squared critical value in the 0.05 column, that means there is a less than 5% chance we got this outcome due to chance alone. Okay, so this may be a little bit to process. There's a fair amount of complexity to this, but basically, if we have a huge chi-squared value, there's a very small chance of actually winding up with this outcome we have up here. So this distribution of observed individuals simply by random chance. So this is not really, an even coin toss here. So the other way of putting this conclusion is, so then these genes are linked. Okay? And if you look across this table, you'll see as you go to the right, this is, what's the probability that this happened by chance at 0.001. So our chi-squared value here, 256, is huge. It's actually off the charts here. So what that means is there's a less than 0.001%, or less than 0.1% chance of having this outcome just purely by chance. So this is, not a random event. This is a loaded set of dice, or this is a coin that is not evenly weighted here. Okay? So one of the main things I want you to take away from walking through this set of calculations is that this statistical method is a quantitative way to make a decision about the outcome of this experiment. And it's a way that lets us make that decision without just kind of squinting at the numbers and deciding that, okay, those genes are linked. Because there could be a borderline case where it's not entirely clear. And in that case, this will help us kind of reach the conclusion that, okay, these genes are not linked. Or, okay, yes, these genes are actually linked. And this is a lot to think about, but statistics play a really important role in a lot of biological questions and in a lot of biology research. So it's important to spend some time digesting these ideas and getting more comfortable with them. But at the same time, working through these calculations is kind of a big project. So at this stage of things, I just want you to know that I'm not going to ask you to go through this whole process on an exam. And one of the big reasons is it would just take way too long, and we have a lot of other topics to cover. But I do want you to practice this type of statistical thinking that I've outlined here. So if I give you outcomes from an experiment, this information, I give you a calculated chi-square value, and I tell you what the critical value is, I may then ask you what you would wind up concluding. So if, for example, you get a chi-squared value that's really small, then, okay, there's a chance that this could have just happened through random chance. And in that case, you could say, okay, well, these genes are not linked. But if you get a large chi-squared value that's bigger than the critical chi-squared value, then you'd want to say something similar to what's here, that there's a very minuscule chance of getting this type of outcome just purely from chance. So I would conclude that these genes are in fact linked. Here's another example. So in this case, with this example, you'll notice the observed offspring are 86 for the dominant, 116 dominant recessive, 110 recessive dominant, and then 88 recessive. So here again, our expectation is the same if unlinked, right? Okay. And so if we do all of the number crunching, we wind up getting a chi-squared value of 6.96. And our critical chi-squared value is still the same here. So I want you to think about what you would conclude for this cross. Are these genes more likely to be linked or unlinked? And along with that, for this particular question, I want you to think about whether the two dominant alleles are on the same chromosome with each other and the two recessive chromosomes are on the same, or recessive alleles are on the same chromosome with each other, or whether it's more likely that we have a weird mixture where there's one dominant and recessive together on a chromosome and one recessive and one dominant together on the other chromosome. All right, so a lot to think over, but take your time with it, and I think eventually it will start to make sense.